So we were learning about oxygen hemoglobin dissociation curve. The uptake of small amount of O2 by hemoglobin favors the relaxed R state of its peptide chains. This in turn promotes the uptake of additional O2. This can be graphically represented by a sigmoid S-shaped curve known as oxygen hemoglobin dissociation curve. Oxygen hemoglobin dissociation curve, also called oxygen dissociation curve or oxygen equilibrium curve, is a graphic representation of the affinity of Hb for O2 at different levels of PO2. In other words, it is a graph which represents the relation between the percentage saturation or oxygen carrying capacity of Hb and different levels of PO2 in the blood. It reveals the actual relation between the PO2 of the foam and the formation and association of oxyhemoglobin. Concentration of the first heme molecule with O2 increased the affinity of the second heme molecule. In the same manner, oxygenation of the second heme molecule increased the affinity of the third one, and oxygenation of the third molecule increased the affinity of the fourth molecule. Thus, the affinity of the fourth heme molecule for oxygen would be many times greater than that of the first molecule. When hemoglobin takes up O2, its two beta chains move closer to each other, and when it gives up O2, the beta chains move further apart. It is this shift that determines the shift in the affinity of hemoglobin for O2. The oxygen hemoglobin dissociation curve corresponds to the fact that at 0 PO2, there is no HbO2 in blood. With a rise in PO2, the curve goes up gently at first and then rises more sharply. Thus, with the PO2 of 10 mmHg, the blood may contain 10 to 15% of HbO2. With a PO2 of 20 mmHg, it may contain 30 to 35% of HbO2. And with a PO2 of 30 mmHg with HbO2 content reaches 50 to 60. With a further increase in PO2, the curve flattens out again. With a PO2 of 40 mmHg, the content of HbO2 may reach 70 to 75%. With a PO2 of 50 mmHg, it corresponds to 80 to 85%. With a PO2 of 60 mmHg, it reaches nearly 90%. After that, the curve runs almost horizontally. An increase in the PO2 from 60 to 80 mmHg adds nearly 6% HbO2, and from 80 to 100 mmHg only 2%. Thus, the curve asymptotically approaches a horizontal line which represents the upper limit of saturation. So, curve at normal blood pressure temperature shows the relation between O2 and Hb saturation, PO2 and Hb saturation. Percentage saturation of hemoglobin will be represented on the y-axis, and the oxygenated blood is there, and PO2 mmHg on the x-axis. The sigmoid curve is shown. Some other aspects: curve showing the relation between temperature and Hb saturation. As temperature increases, less O2 combines with Hb. Then dissociation curve showing the relation between PO2 and Hb saturation. Sigmoid curve, normal shift right side will be shown. And PO2, fetal and maternal. Effect of various factors on oxygen dissociation curve. Many factors influence the affinity of the heme molecule for binding with oxygen and altering the shape of the curve. Left side of the curve is a sign of hemoglobin increased affinity for oxygen. Similarly, right side shows decreased affinity, as would appear with an increase in blood temperature, hydrogen ion concentration or carbon dioxide concentration, the Bohr effect. Carbon monoxide has a much higher affinity for hemoglobin than oxygen has. In carbon monoxide poisoning, oxygen cannot be transported and released to body tissues, thus resulting in hypoxia. With fetal hemoglobin, the shift facilitates diffusion of oxygen across the placenta. This oxygen dissociation curve for hemoglobin, myoglobin exists even further to the left. Then transport of carbon dioxide. Earlier we learned about effect of various factors on oxygen dissociation curve. Many factors influence the affinity of the heme molecules for binding with oxygen and altering the shape of the curve. Left side of the curve is a sign of hemoglobin increased affinity for oxygen. Similarly, right shift shows decreased affinity, as would appear with an increase in body temperature, hydrogen ion concentration or carbon dioxide concentration, the Bohr effect. Carbon monoxide has a much higher affinity for hemoglobin than oxygen has. In carbon monoxide poisoning. Oxygen cannot be transported and released to body tissues, thus resulting in hypoxia. With fetal hemoglobin, the shift facilitates diffusion of oxygen across the placenta. The oxygen dissociates, curved hemoglobin, myoglobin exists even further to the left. Then about transport of carbon dioxide, a healthy human adult under resting conditions produce nearly 330 liters of CO2 in 24 hours. The CO2 formed in tissues diffuses to blood. Blood transport it into lungs where it diffuses out from blood and escape to the outside during expiration. Blood transports CO2 from tissues to lungs in three ways: as carbonic acid, as carbaminohemoglobin, as bicarbonates. Then transport of CO2 as carbonic acid. In this case, CO2 reacts with plasma water and forms the weak carbonic acid. This occurs in tissue sites. On reaching the lungs, carbonic acid dissociates and releases CO2. In man, about 5% of the CO2 is transported in this manner. The remaining 95% is transported by RBC. CO2 plus H2O gives H2CO3 plus gives H2O plus CO2. The formation of H2CO3 in blood plasma is influenced by the solubility coefficient and tension of CO2, PCO2, and the internal temperature of the body. In many invertebrate species, the reaction is catalyzed by the enzyme carbonic anhydrase present in their blood plasma. In vertebrates, this enzyme is present in the RBCs, so the formation of H2CO3 in plasma is a very slow process in vertebrates. Then transport of CO2 as carbaminohemoglobin. In this case, CO2 first diffuses from plasma to RBC, then it reversibly combines with the amino NH2 group of the globin part of hemoglobin and forms carbaminohemoglobin. The formation of carbaminohemoglobin is greatly influenced by the high CO2 in the tissue capillaries. So also its extent depends on the availability of NH2 groups and the pH and PCO2 of the blood. On reaching the pulmonary capillaries where PCO2 is very low, carbaminohemoglobin dissociates, releasing CO2. This dissociation is catalyzed by carbonic anhydrase. The CO2 soon diffuses to pulmonary alveoli and then escapes to the outside. In man, about 10% of the CO2 is transported in this manner. CO2 plus HbNH2 gives HbNHCOOH, HbNH2 plus CO2. Then transport of CO2 as bicarbonates. Isohydric transport.